Okay, so it seems to be more, either people who have no experience or people who have some experience but don't use it very often or have questions. Interesting. Okay, that's good. So I think my choice of starting with a presentation about OMERO itself is probably a good idea. Okay, I think we can probably start. So quick introduction. My name is Erick Ratamero. It's not reflected here in the presentation because I am very shamelessly just taking a presentation from the OME folks themselves because it's a good introduction to OMERO. So my name is Erick Ratamero. I work at the Jackson Laboratory, which is a biomedical research nonprofit based in the Northeast in the US. I've been working with OMERO for maybe 7 years now. I started just as a person running an OMERO server for a medical school. Eventually, yeah. Doug thinks the command line makes him nervous. That's cool. We're going to look a little bit at that. Yeah, so I started just being like the person who was the admin for an OMERO server. Eventually, when I moved here for this job, I started developing a bunch of functions for doing common things in Python. And that turned into ezomero, which is the main thing we're going to talk about today. And I ended up developing a few other OMERO utilities around the main service. My background is in computational physics, so my PhD was in computational physics, and I kind of got into microscopy through the window. I got a job to do image analysis, even though in my interview I told them that I had never worked with images in my life before. They still decided to hire me, so that's how I ended up doing imaging things. And yeah, I've been doing it for about 7 years, and it's been very good. So that's me. I'm going to talk a little bit about OMERO itself because the level of familiarity seems to be a bit variable here. We have everything from like people who have never seen OMERO or just curious about it to people who have used it before, people who are familiar but have questions or worries about specific things. So let's start talking a little bit about OMERO itself. As I said, I'm just stealing this presentation from the OME team themselves, so the team who develops the core part of OMERO, which I'm not part of. Okay, so OME itself, so that is also open microscopy and OME are kind of like interchangeable terms. OME stands for Open Microscopy Environment. They are a team and a consortium of institutions that developed this common data model for microscopy data. You might have used OME TIFFs in the past. OME TIFFs use that same data model. And the central idea of OME is mapping everything into this one data model and making that the center of your data analysis and data management tools so that everything can talk the same language and use the same concept. So essentially, if you could have all your raw data translated into this common data model, that means that all your analysis, all your visualization, all your process data could rely on that data model and make everything easier. So as I said, you might have used OME TIFFs before. OME TIFFs use this data model. It's also what's behind bioformats, which is also something you might have used in the past to open images in Fiji or whatever utility you use for image analysis. And OMERO is just essentially a layer on top of that data model. So at the center, it's still that same data model. The OME data model is still there. What OMERO does is you import data into the OMERO server. That server will extract everything it can that conforms to the OME data model. It's going to store your data in some secure storage. It's going to parse all the metadata that it could get from the OME data model into a database. It provides search, it provides analytics, and it's all running in a server that you can interact with through the internet. or, you know, it can be accessed directly from an acquisition computer or a processing computer. So essentially, you just put your data somewhere safe. You have all your metadata parsed there. You are able to search your data and analyze it. And you can access that data from anywhere you are. As long as you can talk to the server, you can find your data. So none of like 10 different external hard drives that you need to plug one by one until you find where your data is. None of that. Your data is always in the OMERO server. You can search it, you can annotate it, you can analyze it, and you can access it from anywhere you can talk to the server. So an important thing here for what we're going to do later is that OMERO is a server with clients. So OMERO itself is a server, and everything that you do on top of that server is going to be a client. It's going to talk to that server and get things. So for example, when we look at the web page, that is the web viewer for OMERO, that web viewer is a client itself. It's talking to the server in the background. And the things we're going to be doing with Python are also going to be talking to that server using Python. Logistic point, if you have any questions at any point, feel free to either put them in chat or in the Q&A panel. I will try to keep an eye on both. Yeah, I'm happy to answer questions at any point. You don't need to wait for me to stop or anything like that. Just put the questions there. I'll get to the questions as they come in. Okay, so I mentioned bioformats. Bioformats is also a thing developed by the OME team. You might have used bioformats many times before, either knowing or unknowingly. It supports reading more than 150 image formats. So essentially anything that your microscope spits out, bioformats will be able to read. It can read both the pixel data, so the actual pixels in your image, and the metadata, so whatever the microscope vendor decided to put in that file format. If it fits into the OME data model, bioformats will be able to read it. And that includes all kinds of different images, so 5D images, high content screening, whole slide images with tiles, everything. It's mostly geared towards biological images, as you might imagine by the name bioformat, but it also does some medical imaging. So it has support for like nerd files and DICOMs, if that's your world. And it also supports general images like TIFFs and PINGs and stuff like that. And that's what OMERO will use to take your original data, parse it into the OME data model, and store that metadata in that data model. So yeah, as I mentioned, the metadata is stored in an actual database. It uses Postgres, if that means anything to you. It's both the acquisition metadata, so this is what the microscope vendor puts in their file format, but it's also things that you as a user can add to your image. So for example, any kind of user added annotations, but also regions of interest, segmentations, masks, things like that. And it's all stored in that same database. It's searchable, it's accessible from anywhere, and so on and so forth. OMERO is a very nice tool in terms of being able to integrate with other things. So if you use Fiji, you can access your images from OMERO in Fiji. Same thing with QPath, all kinds of Python things, Elastic. If you're using stuff from Visiofarm, Napari, R, MATLAB, Cell Profiler, all kinds of things. So the dream of OMERO is this thing where your image is in a server, stored safely. You don't need to worry about it. And then whenever you need to do anything with your image, either you just use a web interface to do it, or you use an analysis tool to connect to the server, do things with your image, and then push the results back to the server again. That's kind of like the dream of OMERO is that. The reality, it's always a little bit more difficult than that, but that's the concept, that's the vision. And for many of these things here, that's actually how it works. So if you have whole slide images, you can open them in QPath, do whatever you want in QPath, saves the results, the segmentations, whatever, back to OMERO, and it all just works. instead of going through the rest of this presentation, I'm just going to show you the web viewer for OMERO. Another logistics thing here is that I'm going to run this workshop mostly as a demo and a tutorial rather than a kind of like hands-on thing. And I consider doing it as a hands-on thing where each of you could actually open a Jupyter notebook and write code that is going to interact with the server. The difficult part would be having a server that everyone will be able to access, that everyone would have credentials for, and especially not knowing exactly how many people were going to show up. It was very hard for me to gauge, if I am going to deploy a server, say in the cloud for everyone to use, how big does that need to be? How many people are going to be accessing it? So eventually I opted for just showing the things instead of having you work with me through these things. I will show you, actually, let me put it in chat right now. The GitHub repository for everything I'll be using is here. It's just a few Jupyter notebooks. So if you have access to an OMERO server, I'm going to show you how to use these things later, and feel free to just work through these on your own time. If you have any questions, feel free to reach out to me. Yeah, hopefully the instructions I have there for installing and everything are sufficient. But if they're not, feel free to reach out to me as well. So yes, instead of continuing this presentation, I'm just going to show you the web interface for OMERO, just so you have an idea of what this actually looks like in practice. And here I'm using my, the internal Jackson Laboratory dev server. So that's why dev is here. It's called CT OMERO dev. You are not going to have access to this because it's internal. It's inside our network. I just want to show you what an OMERO server looks like. So here we're looking at OMERO Web. OMERO Web is a browser view of your OMERO server. And following that paradigm that I showed before, it is a client, right? This is not the server itself. This is just a web interface that talks to the client and shows you things. So the classic OMERO web interface here has 3 columns. On the left here, you have this kind of like tree diagram that you might be familiar from, Windows or Mac or anything like that. You just have, things that look like folder structures and images there, and you can click them individually if you want to. Here in the middle, you have thumbnails for each of your images. And then here on the right, you have metadata. And metadata can mean many things here. It can mean, for example, when this was imported into the server, dimensions of the image, what kind of pixel type, pixel sizes, how many Z sections, time points, channels. If you have channels with names, they will show up here. How many regions of interest in that image. It can also be things added by the user. So for example, here, you can just add a key value pair here that says that, for example, this image was treated with mitomycin A at 10 micromolar, for example. If you change here at the top to the acquisition tab, then you get the original metadata that came from the microscope. So this can be, in this case here, these images are example images, but they will have things like, let me see if they have any of these here. These don't. Let me see if the other ones do. They'll have like information about the microscope, right? Like this is a Zeiss microscope, this model, this was the magnification, NA, immersion, correction, and so on and so forth. So all the things that OMERO was able to kind of like slurp out of the original file from Zeiss and map into the OME data model, they are going to show up here. So you have access to a lot of the original metadata from the microscope. And then finally, at the end here, you have a preview tab. And here, for example, you can very easily turn on and off channels. You can adjust brightness and contrast, even using a histogram if you want to. So I'm just going to play around here with a little bit, all the things that you expect to be able to do in an image viewer for microscopy images. You can save these. For example, if I save these rendering settings here, you'll see that it will update my thumbnail here as well. If you want something a bit more potent than just this preview tab here, you can open this image with iViewer, for example, which is another client that talks to the OMERO server. And in this case, It's a bit more advanced viewer. It has the same things that the basic preview had, like the histogram. But here you can also, for example, change the lookup table for each individual channel. You can draw regions of interest. So I can draw a rectangle here, for example, save it to my image. This rectangle is not useful, of course, but for example, if you wanted to get like each individual point that you have in this image here, you could just click on each one of them. And all of a sudden you have a bunch of points you can save to your image, and they're all there. So if you're just doing, for example, annotation on your image, finding regions of interest, things like that, you can just do all of that in your browser. None of this is in my computer, right? Like I'm just talking to the server and it's all in the server, and it's just gonna be there. If I go to a different computer tomorrow, I will just log into the same server, everything will be there. Yeah, so that's a very, very, very quick summary of OMERO. One thing that I will also mention, because it's going to be relevant when we start writing code here, is that OMERO doesn't do folder structures really. So you cannot, for example, inside this folder here, put another folder. That is not a thing in OMERO. OMERO gives you two levels of quote unquote folder structures. It gives you projects, which are the blue ones, and it gives you data sets, which are the green ones. If you need any more organization on top of that, there are options like, for example, you can add tags to your images. So for example, if you need any kind of, subset of these images here, you can just add a tag to those subset. But the idea is trying to break people from the idea that organizing things in folders is equivalent to data management, right? These are two different things. You can do data management without folders, and folders are not data management. So OMERO is very adamant about this specific structure. It's also what's in the OME data model. The OME data model doesn't have anything more than projects and data sets. That's it. So this is going to be important when we actually start looking at code things. So here, I'm going to stop for a second and ask if you have any questions, any curiosities, anything you are interested in. If you've never seen OMERO before, I'm sure you will have questions about like how does this work and what things it can do and it cannot do. So feel free to ask those now before we go into code. And I'll also take that to drink some water. All right, questions? No questions? Okay. Conventional file size limits for individual images. There are none. We have in our internal server, we have lots of cases of like individual whole slide images that are multi-gigabyte in size, and they just work. OMERO will use, so for very large images in general, what you do is you create pyramids. If you're familiar with pyramids, essentially you create multiple versions of that image that are downsampled, and you do that multiple times until you get something that's manageable, so that when someone opens an image, they're opening the very low resolution version of that image. And then when they zoom into one part, then you load the high resolution version of that part. OMERO does that for you if your images don't have that. So you always end up with something pretty manageable regardless of the size of the images. The one part that can be annoying depending on your file format and how big your images are is the import process, because OMERO will want to do things like, for example, if I go to my preview panel here, you will see that OMERO has calculated here what's the minimum and maximum value of the red channel, right? And to do that, it had to read the whole image. It had to calculate a minimum and maximum for the whole image. So if you have a very large image, these calculations can take a while. But these are part of the import process. As soon as the image is imported into OMERO, it's no issue at all. Okay, yes, Alexander, I can do that. Yes. I don't think it is in the list of things we're going to be doing. I think we're going to be posting a key value pair over the API, not deleting it, but I can show you that as well. Yes. So Doug is right that like sometimes you have like one very large file or one very large plane, for example. And it's a good idea to pre-convert them to something pyramidal like an OME TIFF before you import. If you don't do that, then OMERO will try to calculate the pyramids when you import. And that is a process that is one, very, 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 very time consuming and processing heavy, two, very prone to errors. And this is something I have faced in the past as well. So yeah, if you have very large single plane images that don't have pyramids, it's a good idea to convert them to something like an OME TIFF before importing. Yeah. All right. I will jump into code things if there are no more questions. That's also a good point to ask, how familiar are folks with Python in general? Does it support 3D data? It supports 5D data. It supports anything up to 5D. So this here, for example, is there are 23 time points, 2 channels on this image here. So this is a 4D image. So if I open this with iViewer, for example, I can just press play here at the bottom, and you're just going to see each individual time point and both channels in the same image here. Yeah. All right. Now, if you don't know any Python, what's coming next might be tricky to follow. If you know some Python, you're probably going to be okay. I'm going to tab over here. Actually, before I do that, this is what we're going to be using next. That's a good point from Doug as well. Video formats can be tricky. I think OMERO can do .avi, .mov, but only with specific codecs. So it's not every codec that works. So if you have like a regular video that you took with your phone camera, probably not going to work. If you have something that was generated, say, with a raw codec from actual scientific equipment, it will probably work. So Bioformats has a list of formats that's out there. Let me find it. And it's a very good reference material for knowing like what kind of thing can I put into OMERO. Let me put it in chat. So anything that's in this list there can go into OMERO. So for example, if you go into the AVI files, which are under AVI, let me show you here. You will see that, for example, these codecs are supported. So it's just MSRLE, MSV1, RAW, and JPEG. So if your video was generated with that, you're okay. If it wasn't generated with that, then it's not going to work. But yeah, they have a very big list of file formats that you can use bioformats with. And anything that bioformats can do, OMERO can do. Okay, before I jump into code, this is what we're going to be using. It is called ezomero. And ezomero is something that we developed in-house at JAX. Basically, there were lots of things we wanted to do in Python to talk to an OMERO server that were kind of cumbersome or annoying to do using the basic Python API that OME provides. So we started writing like small functions to do specific things. And eventually we just said, you know what? We should just make this public. Other people might also find use from this. And that's where we are. Documentation is here if you're interested. So if I just go in here, you know, each function has its own documentation and so on. If you know, Python technical documentation, all of this is going to look very familiar. But yeah, ezomero is not an API by itself. it builds on top of the existing OMERO Python API, and it's just purely for convenience. The idea is making people's lives easier when they're writing Python code that talks to OMERO, which is why it's called ezomero. Which also means that not everything you can do with a Python API for OMERO is in ezomero, right? So for example, Alexander's question about like, how do I delete annotation using the API? That is not in ezomero. We did not write a function to delete things. I think so. I think it's not there. But for example, a function to create a new annotation is there. And it is a simpler version of what you would need to do if you're just writing things with the basic API. Anyway, I've talked a lot about that. Let's hop in to actual code. Okay, before we actually start looking at what ezomero can do, I just want to give you like a very quick example on how to get an image from OMERO using two different methods, actually. It's just kind of like a teaser for the things we're going to be doing, just to show that it's actually pretty easy to do, like just get an image with the scaffolding that we built in ezomero. So in addition to the two methods I'm going to show here that use two different APIs, there is also a command line interface for OMERO that can do a lot of things. And then there's the actual OMERO Python API. And that's essentially, you can do anything with that. But yeah, if you're doing something like a bash script or trying to automate something, the command line interface is also very powerful and very useful. Okay, first things first, if you're going to be using this repository in the future, trying to talk to an OMERO server you have access to, you will find there, let me put the repository on screen as well. You will find there a config template.py file. And this is, you will need to give like credentials, the address to your server, and the address to your OMERO web interface. So in our case here, it would be the actual website. Of course, if you're doing anything serious, please don't put your actual password in a text file. That's a very bad idea. But just for a quick example, or using, you know, a throwaway account or anything like that, or just inside your internal network, that's not a huge issue. But if you're doing anything serious, please, there are better ways to do that. I'm happy to talk to you offline about how to do it and not needing the password there. Actually, I'm going to show you that you actually don't need that password. Okay, so in my case here, what I did was I took that template and I generated an actual config.py file here. And in my case here, I don't mind you seeing that, you know, my username is user1 and my password is OMERO, because this is a dev server. It's inside our internal network. None of you will have access to it. And this user doesn't have any privileges anywhere. So it's not an issue. But this is essentially the amount of configuration you need to do to set up a connection to your server. So in Python here, I'm just going to import all these things I just created here. So these variables. So I'm just importing from the config file here. And actually, let's try not importing the password, just as an example. And then I'm going to import a couple of functions from ezomero. So I'm just going to import the connect function and the get image function. I will also need matplotlib to actually show the image. And then I'm going to pick an image here to display on my screen. So let's pick this one. This one is very distinctive here. It has like this one cell that is in metaphase. It's very nice. And here in this case, I just want to note what the image ID is. So the image ID here is 57813. So I'm just going to create a new variable here with that image ID. Okay, so the process to get this image from the server has exactly two steps. The first step is connecting to the server. and I'm going to omit the password there. You'll see that I will be prompted for the password if I do that. So I'm just going to run this. It's just going to ask me what the password is. My password is OMERO. It's going to do things. No, do not save that password. And now if I print this connection object that was generated here, you will see that it is actually a gateway, Blitz gateway object, which is an internal OMERO type. But that means the connection has been successful. So now we have a connection to our OMERO server, and the connection is stored in this variable here. Now, to actually get the image, it's one extra step. So I'm just calling the getImage function that I imported here. I'm giving it the connection object that I generated. I'm giving it the image ID. And I'm specifying that I want the image to come back to me in this order of dimensions. So X, Y, Z, channels, time, which is normally what you want most of the time. So if I run this, it's going to retrieve my image from the server. And now the image, I have an image object that is an internal OMERO type. So you see that it's an OMERO gateway image wrapper type, but I also have pics, which is a NumPy array. And if you do any Python with images, you are familiar with NumPy arrays. So for example, I can do this. And you will see that the image I got is 305 by 240. And we can double check here and see that it is 305 by 240 and see that it had 23 time points and 2 channels. And here we have 2 channels, 23 time points. And then if I want to see what the first channel, first time point looks like, that's what it looks like. So this is the red channel on this image here. And if I just print pics, you will see that this is just like a very large array with the actual pixel values from your original image. And these are the actual pixels. This is like literally the stuff that came out of the microscope. The actual values is what you get here. So summarizing here, the thing I did was essentially I created a connection. I ran a get image with the image ID, and I got everything I needed. So if I wanted to do any analysis in Python, for example, I could just use the pixels NumPy array that I have here to do whatever operations you want to do with scikit-image or OpenCV or whatever you want to do, generate whatever you want. And there you go. You would have your analysis. Okay, before I move on, I want to show you how to do a very similar thing, but using a different API. So this here is just under the hood. It's just using the basic OMERO Python API. An alternative way of doing this, if I want to do, okay, I can show you what the getImage function looks like in Easy OMERO, and that might give you a decent idea of how complicated it would be. So this is getImage here. It starts on line 28. The actual code starts on line 110. And it goes to line 327. It would be like, 200 lines of code. But of course, this is like for all possible cases and it's doing a lot of, checking for types and things like that. So I think if you were doing it in a very basic way, it would be maybe like 10 lines of code, which instead we have 1 line of code, which is easier. Maybe 10 is a bit amazing. Maybe like 7 or 8. That's probably the kind of like the ballpark for just getting a basic image. It would be like maybe like 7, 8 lines of code. But that's something you want to do very often, right? Like you don't want to write the same 7 or 8 lines of code every time you need to get an image. Eventually, if you're writing code that deals with OMERO, you would have written a function that gets you images. And what we did was essentially just make our function public to everyone so that they don't need to write it. Okay, so I'm going to show very quickly how to, I will also get the same image, but I will do it from the web interface instead of getting it from the server directly. And that's a very subtle difference that is important if you're dealing with OMERO. So here I got the images from the server. The pixels I got here are the pixels that were stored in disk by the microscope when the image was generated. The alternative I'm going to do here now is I will get the image from the web server. So from the web interface. Okay, I didn't, okay, that's because I didn't import OMERO Pass here to test the thing. I don't need this, but I do need to rerun it. And hopefully now I can run this. Yeah, I'll get some warnings, but that's fine. The warnings are because our internal server here, the SSL certificate is self-signed. So, you know, it will complain about things, but these are just warnings you can ignore. So what I'm going to do here is I'm going to use the web interface to get a rendered JPEG of that image. So if you just want to look at the image, this is fine. You're getting a JPEG that is literally this image here as rendered in a JPEG, right? So it's going to take into account your rendering settings that you have here. So like your brightness and contrast, which channels are on and off. It's going to take all of that into consideration. So if you just want to look at the image, you can just go straight to the web server, and you saw that it was essentially instantaneous. It just popped up. It's much faster. It's much lighter. If you just want to look at the images, this is totally fine. You should not do analysis on this here, because this here is not your actual pixels that you got from the microscope. This here is a JPEG. So you see that, for example, even the scale is completely different, right? So these are like values between 0 and 255. The ones here are like in the 30,000s. So this is fine if you're looking at the images. This is not an image you should use for analysis. But we got it straight from the web portal instead of going to the server itself. So I just wanted to make sure that, you know, you saw both things so that you knew both things were possible and both things have their own uses. All right, before I dig deeper into things, any questions? We're doing more than okay on time, so feel free to ask as many questions as you want. Okay. I'm gonna go to a new notebook here, just so we start fresh. I'm going to focus on the essentially the first part of this, right? The parts of the ezomero library that talk directly to the server instead of talking to the web interface. If we have time and people are curious, I can go back and look at the parts that would talk to the web interface instead of the server. Okay. Right. So just a quick summary on what this is and what it looks like. So ezomero uses, as I mentioned, the OMERO Python API to talk to the server. That Python API in turn uses a thing called ICE to talk to the server. And ICE is what's called the remote procedure framework. So it's a way of running things remotely in a way. So this Python API is generating things that are talking to the server and saying, hey, please run this for me and give me the results. And that's what ICE is doing. So that's essentially the language the API talks to be able to talk to the server and ask for things. So we're essentially like 2 or 3 layers abstracted from the actual server in many ways. Which is great because that means we can do things in a more concise and less verbose way, but it's something to keep in mind that we are a few layers away from the actual server. And that's particularly relevant if you run into issues, right? Like you need to start figuring out at which of the layers is my issue actually at. And that's not always trivial. Okay, so the first thing I'm going to do here is I'm going to import some stuff. I'm going to import the config things again. So it's just, username, password, host, port. I'm importing matplotlib again to be able to see images, and I'm just importing the whole ezomero library because it's easier than importing the individual functions here. So I'm just going to run the imports. And then first thing I'm going to do again is I'm just going to create a connection again. So I'm giving it the username, password, what port in the server, what the server is. Here I'm saying I don't mind which group I'm in. I didn't talk about groups, but of course in OMERO we have this concept of groups and users can be multiple groups. It's essentially a way of controlling privacy and properties of images so that, if you have images that you don't want everyone else to see, only people in your group are going to see them and so on. So like permissions, privacy, and so on. So if I run this, I get, again, this Blitz gateway object. So that means a successful connection. So the first thing I'm going to do here is I'm just going to, in here, I'm not using ezomero at all. I'm just using the connection object itself. You'll see that that connection object already has a lot of stuff. So in this case here, I just got the current user for that connection. I'm just going to print some stuff about the user. So what's the user ID, username, and the full name. And yeah, in this case, there is everything in OMERO has a unique ID. So this user has unique ID 205. The username is user1, as you might remember from my config file here. And the actual name of that user is training user, user1. Of course, this is like if you were using an actual OMERO server, it would be the actual person's username and their actual name. And then you can do more things like, for example, let's see which one of these I actually want to. So for example, you can check which groups is this user part of. So here, for example, again, I'm using the connection object. I'm getting which groups that user is a member of. For each of those groups, I'm getting the ID for the group. Actually, this is inverted, isn't it? This should be ID, and this should be name. And then I'm getting the current group that the connection is using, and printing what that is. So in this case here, I'm only a member of one group, which has ID 103, and it's called training. And that's the current group my connection is in. So if you were a member of multiple groups, for example, you can switch which group your connection is, what they call it the context. So like the group context. You can think of it of, for example, imagine you as a bioimage analyst, you're doing work for 3 different labs in your institution, and you are a member of the 3 different groups. If you want to grab an image from group A, you need to make sure your connection is in group A. If you want to grab an image from group C, you need to make sure to switch groups from A to C before you do that. So this is all something that you can do in OMERO as well. Okay, so as a refresher, I'm going to do the get image again. So that's the first thing I'm going to do here. What was the ID I was using? I was using this one. Let's do the same one for consistency. Again, here I'm just using ezomero get image. It gets me a pixel array and an image object. I just need to give it the connection and the image ID. And here we're going to be printing what the shape of this NumPy array is and showing the NumPy array itself. And this is going to look very familiar. You see that this takes longer than getting the JPEG that we got here, down here, because you're actually getting all the pixels for this image. Which is something you need to keep in mind sometimes if you're dealing with very large images. There are things you can do in get image here to get, for example, if you have a pyramid, you can specify which pyramid level you actually want to get. So if you're dealing with a very large image, but you only need a low resolution version of it, you can say pyramid level equals 4 or whatever, and it's going to give you a higher pyramid level. In this case here, we're just getting all the pixels for that image and showing them here. And the shape looks like this, same as it was before. Of course, we had to actually go here in the web interface and check here on the general tab to actually find out what the image ID was, which is one of the essential parts of getting the image. You need to know the image ID. If you're writing code, normally you don't want part of your workflow to be open the web browser, find the image on that data set and copy and paste the image ID. You want to find these IDs in your code. So in our case here, we can go one level up. For example, we can look at the data set ID here. And in this case, it's 1281. And you can, for example, do data set ID is 1281. ID list is ezomero.get image IDs. And it's just going to be your connection object. Your data set is data set ID. And then you can print a list if you want. And if you run that, what you get is an actual Python list with every image ID on that data set. And that includes our good friend 57813, hopefully, because otherwise it will look bad for me. It's here. Okay. Yeah, so if you're writing code, for example, that needs to do this, does that also work for plates? Kind of. For plates, you would get from a screen, for example, you would get plate IDs. From your plate, you would get well IDs, and so on and so forth. They're like the hierarchy is slightly different, but yes, this all works for plates as well. In fact, if we look at our code here, let's see what we have. We have get plate IDs is in ezomero. Get well IDs is in ezomero. Get plate acquisition IDs is in ezomero. Get well ID is also in ezomero. And in this case, you just give it like a row and a column and you get an ID back. Yeah. So some of this has been contributed by other people at this point, which is very nice. So if I'm not mistaken, the plate acquisition IDs is a pretty recent addition that was, someone needed this and they wrote a function and put it in ezomero, and we just merged it and it's in the library now. Yeah, so for example, and this, like, we have getters for lots of things, right? So for example, I can do something like this. I'm just going to copy paste it because it's easier. You can do get map annotation IDs, for example. And in this case here, you need to give it a connection. You need to say it's an image because the map annotation can be on anything in OMERO, essentially. So you can have a map annotation on an image, on a data set, on a project, on other annotations. It's a mess. So in this case, you need to say it's an image you're looking at and you give it the image ID. And if I print that, you'll see there are 3 map annotations there. And for example, if you want to see what's in one of them, you can use a print map annotation. You give it the connection object and one of the IDs we got here. And you will see that it gives you the map annotation ID, what namespace that annotation exists in, and the actual contents. Yes. So of course, the thing I didn't go one step further here. Like how would I get the dataset ID, right? So of course there is a get dataset IDs that you can pass a project to, and there is a get project IDs that is just going to give you all the projects that a user has access to. We also have something like this that doesn't return anything, but it's just like if I want to know which projects I have access to, you can just print those. And in this case here, it doesn't look super useful, but it's mainly because this group, this training group that I have here, just has 50 identical users, actually 30 identical users. And they all have access to each other's data. So I just get 30 identical training data projects that have different IDs. Of course, if you're doing this in a real OMERO server, you actually get something useful, and it tells you which projects you have access to and its IDs. All right. I will pause for a second to see if there are any other questions. Okay. Of course, I haven't shown you every single one of the get functions that we have here in ezomero. Um, but this is all in the documentation. So if you have anything that you would like to do with ezomero, you can look at the documentation, see if it's there. If it doesn't exist and you would like to contribute back to ezomero, we are very, very happy to accept external contributions to ezomero. And I will show you here that we are at this point at 10 different people who have contributed code to ezomero. It started as 2 people writing a tiny library. And at this point, there are 3 other people at Jax who have contributed, but there's also, Will Moore, who works on the core OME team, Tom, who works in Düsseldorf, Julio, who's in Montpellier, um, Dresden, lots of people from Germany seem to like OMERO, so we got a lot of contributions from Germany. But yeah, at this point, the code has a lot of things that were not written by us, which is great. It's a healthy place to be with the library. And we're always happy to accept contributions from people. So if there's something that you would like that's not there, even if you don't know how to write yourself, feel free to just open an issue on GitHub and say, I would like this to exist. And I can't guarantee you that we'll make time to implement it, but it's good for us to know that the demand exists. Okay, so now let's do the other side of this, which is creating things in the server instead of just grabbing things from the server. It's also something that we try to make it as easy as possible. So in this case here, for example, if I want to create a new data set, it's just going to be ezomero post data set. You get the connection object as usual, get a name. This is a data set name. And you get a project ID for the project that you want to create the data set in. So I want to create in this training data project here that has ID 941. So I'm just going to say 941. So I'm going to run that. I'm going to print data ID here, just so you see that what we get back is a data set ID. And if I go here and I refresh my page, you will see that now there's a data set called, this is a data set name with ID 1403, which is the thing we got back. So if you have any workflow where, for example, you need to create a data set and then create images inside that data set, it's a one-liner to create your data set, you get the data set ID back, and then you can immediately create your images inside that data set, which is what we're going to do next. And to do that, there's a little bit of copy paste I'm going to do here just because it's NumPy boilerplate. I'm just generating one single image that is 1,000 by 1,000, and it's all random values between 0 and 1. So I'm going to run that. If you look at image, it's exactly what we expect it to be. It's a mess of values between 0 and 1. And now I'm going to create an OMERO image. Actually, before I do that, let me show you that the type of this image is a NumPy array. So we're just going to take that NumPy array and create an OMERO image with that pixel data. So I'm going to do image ID is ezomero post image connection object as usual, my image array, my name for the image, random image, for example, and I'm going to create it in my new data set. Oh, it's data set ID, sorry. And it's done. It's going to give you a warning because depending on how your admin has set up your server, they might not want you to do this. There are very specific technical reasons for that that you probably don't need to worry about, but it's just a warning we always put there because we've had situations where OMERO admins were angry that people were creating images like this because they are actually stored in a different folder in a different way and so on. So just something to be aware of. So if I refresh my page here, you'll see that now in that data set, there's a new image called random image. It is 1,000 by 1,000. And if I open it, it might not look like much. It might just look like gray because it's actually being interpolated. And this is something I had last time. I'm not sure exactly why it doesn't show me right when I don't interpolate. But you can see from the thumbnail here that it's actual like white noise and not just like a block of gray. And in fact, if you want to double check, you can do ezomero.get image connection image ID, and let's do XYZCT equals true. And if you look at the pixels, you will see that they're not all the same value, right? It's actually the same pixel values that we had here. So it ends, for example, in the 09104. Yeah, it is exactly the same value as you would expect. So again, from a NumPy array to an image in the server that is stored safely, associated with the data set and everything, one line of code. You just post the image and that's it. You can also add things to the image itself. So for example, if I want to add an annotation to that image, I can do ezomero post map annotation connection. And again, map annotations can be on anything. So I need to specify it's an image and image ID. And then it's a dictionary, a Python dictionary. So if you know your Python, you know that it looks something like this. And I'm just going to do something like this. Do I need anything else? Trying to remind me. Okay, I do need a namespace for the annotation. Sample namespace. And I do that. It tells me that that object is actually on group 103, which is correct. I don't know what that print is there. Something to look at. If I refresh my page here, you'll see that that image now has a key value pair on namespace, sample namespace with values test1, test2, 2, which is what we had in our dictionary here. So one of the main reasons we created ezomero is that, for example, if you try to create a map annotation using the basic OMERO API, you need to create like a map annotation object, which is very OMERO specific, very cumbersome. Yeah, as Alexander is saying, it's like this is way more convenient. because this takes a base Python type, which is a dictionary, and just does the thing instead of having all the steps of. So for example, I can probably show you what that post looks like here. So you actually need to create like a list, like I think, yeah, a list of lists on the right way, and create a map annotation wrapper and set the namespace to be that string, set the value to be this kind of like list of lists with two things. So it takes all of that annoyance away, and you just give it. Normally, if you want a map annotation in Python, what you want is a dictionary. So we just made a thing that takes a dictionary instead of taking a weird map annotation wrapper. And yeah, one line, you have your annotation there, and it has exactly the stuff you want. You can also create regions of interest using this. And for that, we defined our own types here that live inside ezomero.rois. In this case here, I'm creating a point and a rectangle, for example. So I'm going to create a shapes list here, because one region of interest can have multiple shapes in OMERO. So create a point. So that's going to be a capital P point, x equals 10.2. I don't know, I'm just picking numbers. X equals 75.3. Sure. I'm going to append that to my shapes list. Then I'm going to create a rectangle. Again, it's a capital R rectangle. And for this here, I'm actually going to copy and paste, because I decided to create a rectangle with every single thing you can have in a shape. So it has X and Y values. It has a width and a height because it's a rectangle. I'm creating it at the Z slice 0. It has a label associated to it. It has a specific fill color, which is an RGBA. So this is going to be red-ish with some transparency. It has a stroke color, which, you know what? Let's make it green just for fun. RGB. So it's going to be filled with red-ish quasi-transparent things. The line around it is going to be green, and it's going to be 2 width for the line around it. So I'm going to append that to my shape as well. Shapes, append, rectangle. And then having my list of shapes, the actual creation of the ROI is just post ROI. Connection, image ID, shapes. And if you want to have a name for your ROI, you can do that. And that's it. You get back an ROI ID. If you go back to OMERO here, and I open this thing with iViewer, you will see there is one ROI here on the right. This ROI has two shapes here, as you can see. Let me zoom in here. As you can see, I don't know if you can see the green thing around the red, but that's okay. There's a point. There's a rectangle. The rectangle is slightly transparent-ish, which you can't see very well because the background is just gray. But, you know, it has the label there. It has all the things you wanted with an X and Y, the width and the height, everything correct. So here, the work is just creating the shapes. As soon as you have the shapes to actually create the ROI, it's one line. And this is another great place to stop and ask for questions. Actually, while I'm waiting for questions, let me look for the thing that Alexander was asking about, which is deleting an annotation from the API. Let's see if I can find it. Okay, so the trivial, let me just put that on screen. So Alexander was asking, how do I remove an annotation using the API? So this is a super useful page that I have on my bookmarks because I write a lot of code for OMERO, which is there is a bunch of code samples from the OME team to do common things. So for example, delete all annotations in an object. You need to get the object using your connection object there. It's an image and an image ID. Then you need to list annotations on that object given a namespace. You append them to a list, and then you can just do connection.delete objects. Of course, if you have a specific annotation ID that you want to delete already, you can just do delete object single. And instead of getting a list, you just pass a single ID here. Slightly different if you want to remove annotations from an object, but without deleting the annotations themselves. In that case, you delete the image annotation link instead of the annotation itself. So essentially, you just unlink the annotation from the image instead of deleting the annotation. Okay. So this is what I had in mind for this section. I don't know if there is interest in looking at JSONs. I think it might be overkill. Everything you can do with the JSON API, you can do this way as well. It's mainly, you're going to get like your project IDs and dataset IDs and everything that you can do it directly from the web server instead of talking to the backend server itself. But I don't think we need to dig into that. So I'm going to renew a call for questions. This is a very good time to ask any questions you might have before I decide to go in a completely different direction. All right, going once, going twice. Okay, we have one question. Do you use Docker Compose for deployment? On our actual servers, we use Kubernetes, which is kind of like a souped up version of Docker Compose if you want to think about it that way. We have both our server on-premise that is inside the network and our public server, which you can go to anytime you want. It's fully public. That's for our data that has been published. So it's just images.jax.org. This runs on Google Cloud, but it's also Kubernetes. And they're very similar deployments, one being in the cloud, one being inside our network. I do use Docker Compose for running tests for ezomero. So in ezomero here, if you go to the tests folder, there's a Docker Compose file there. And if you just do Docker Compose up on this file here, you get a full OMERO server running in your computer. And that's very cool. So that's what I do for testing the library. What I do is I run this to create a local server. I run the tests, and then at the end of the tests, I spin everything down. And it's also what I do automatically when there's a new PR on the library, for example. It's literally just doing the Docker Compose up, setting up Python, installing dependencies, running tests, and that's it. I mean, Docker's totally fine. We went with Kubernetes specifically because it allows us to do things like if a container is down for any reason, it can re-up automatically and things like that. And also things like if for any reason you need to have multiple copies of the web server running on top of your backend server, that's also very easy to do. But yeah, I mean, it's totally fine to have a Dockerized version of OMERO running. It's what I use. Like all my testing is on a local Docker server that I'm just running myself. It's a totally fine. It's slightly more annoying on the production level than Kubernetes is more work up front, but it's easier to maintain. Docker is less work up front, but it's harder to maintain. That's the summary. But yeah, especially if you're doing it at like at lab level, you have a Docker like an OMERO server just for your lab. I can see it not being worth the time investment to get a Kubernetes thing up and running. All right, so the basic stuff I wanted to cover, we're done with. If you want to stick around, I will talk a little bit about future things and what's next, what comes next after OME TIFF, essentially. And this is something that you might or might not be familiar with, but it's something I enjoy talking about, and I think it's useful in this context. There's a lot of effort right now and a lot of people putting a lot of time into developing what comes next, what's after OME TIFF, what can we do better to create things that are more efficient, that are easier to use in the cloud, that are easier to use with machine learning. So the things that the problems people have with OME TIFFs right now, how do we solve those? Right now, what people are converging towards, no problem, Doug, have a good one. What people have been converging towards is something called NGFF or OME Zarr. And the idea is using Zarr, which is a file format, as the TIFF equivalent. And the OME Zarr would be a version of Zarr that also has the OME data model metadata. Zarr is very similar to HDF5, if you've ever used HDF5. The way it works essentially is it will take your image and it will break your image into tiny chunks, and it's going to store each chunk as an individual file. That means that you can address each individual tiny chunk of your image individually, which means that, for example, if you're writing an image very fast, you can write all these chunks in parallel. If you're trying to read a bunch of stuff in parallel, you can read all of those in parallel as well. And you only need to load the things that are necessary for whatever you're doing right now. So if you're only looking at a tiny portion of your image, you only need to load like one file or a few files. And that's kind of like the gist of what Zarr is doing. And it has a lot of advantages for things like machine learning, where like your machine learning pipelines for training a model will take a tiny little tile of your image at a time. So you already have those separated as individual files. It's very useful for storing things in the cloud because at that point you don't have an issue with too many files. It is annoying to deal with. For example, if you're trying to create a Zarr file in your personal computer, because computers don't like to have too many files. And this, if you have a large image, it will create a lot of files. So I'm just going to very quickly show you what this looks like, what it's happening there, and give you a flavor of what this is like. So in this case here, the first thing I'm going to do is I'm just going to get some info about the Zarr that is at this address here. And this is one of the cool things about Zarr. You can just have like a URL to where the Zarr exists, and that's going to be enough to actually open the image. So I run this thing here. It's going to go to that Zarr there. There's a warning. It's still running, so it actually needs to go there and check everything. So it takes a little bit to run, but yeah, you get address and you get, this image here has all these pyramid levels. For example, it has 1, 2, 3, 4, 5, 6, 7, 8 pyramid levels. The full scale, full resolution image is 400,000 by 1,000,000 pixels. It's massive. If you try to open this full resolution image, your computer will catch on fire. It doesn't matter how good your computer is. But you have a bunch of pyramid files. So if you go all the way to the lowest resolution, then it's something much more tractable. It's about 3,000 by 7,000. So here, what I'm going to do next is I'm going to open that Zarr, and note one, that opening that Zarr doesn't mean actually loading the data at all. Zarr is very good at only doing things when you need things done. So if you just ask the Zarr library to open a file, it's not going to actually load anything until you actually need to load things. So I'm going to open that same file there. I'm going to load the lowest resolution pyramid level here. I'm going to print the shape, which should be the same as here, and I'm going to plot that. So I'm just going to run this whole thing here. Okay, it didn't import Zarr. That's kind of an important part. Okay, and this is, even being a pretty low resolution pyramid compared to everything else, it's still quite a lot of pixels, and it loaded fairly fast. So this, I think, is a zebrafish embryo. And the original image is massive, right? So, but if you want to look at this image, you don't need to load the massive image. You can just load the much lower resolution version of it just to look at it, which is very cool. If you're only interested in a very small part of that image, you can also just address it directly, right? So I can create an array here that is the full resolution image that is not going to actually try to download the full resolution image all at once, right? But it's going to give me enough information to be able to make a crop of that high resolution. And then only when I actually try to show it, it's actually going to go to the server where the image is, retrieve only the Zarr files that are necessary for this specific crop here, and show me those. So you see that this is actually pretty fast. There we go. So this is just this like 1,000 by 1,000 chunk of the high resolution image. So this is the full resolution, but just for that tiny little bit of the image. And you're able to do that without ever loading the whole high resolution version of that image. And I think that's very cool. Last thing I want to talk about is, how do I create Zarrs? How do I make a new Zarr? And here I have a little example of creating a Zarr from OMERO. So in this case here, I'm using the command line interface for OMERO. I'm just logging in using my username and password as usual. And it created a session for me. Great. I'm giving it an image ID, and then I can just run OMERO Zarr export and pass the image ID. And that's going to create a Zarr on my computer here. So it's going to do all the exporting that it needs to do. And it's doing that for each time point, for each of the channels. So that's why it's doing like all the 0 to 22 here. But there we go. We have a new Zarr file here. You can see it here. It's a whole folder. If I open that, you'll see there are 3 levels here, which are these 3 pyramid levels here. And then if I open one of those, there are the time points. If I open one time point, there are the channels. If I open one of the channels, there's a single file there, essentially. So essentially what it did, it was like it pulverized this whole thing into a bunch of tiny files that are not super useful if you're dealing with them on your computer. It's super useful if you're dealing with them on the cloud, for example. So here, for example, I can open that, that new Zarr I created. Here I'm taking the full resolution version of that, printing the shape, and then I'm just doing some matplotlib magic here to plot each Z, or each time point, actually, I think, because this has time points and not Z slices. So instead of Z, imagine it says T here. Actually, I can make it say T instead. So yeah, very quickly, you can just, because Zarr has kind of like pulverized this into a bunch of files, it's very easy to get each individual time point here. And you can see that at no point I needed to worry about the individual files inside this folder. I just told it to open the folder. And that essentially gives you kind of like a logical 5D array here, well, 4D array in this case, just by opening the whole folder, Zarr knows to look for the right things in the right places. And if you plot the shape, if you print the shape of the whole folder, it will give you the right shape for the whole collective of images instead of taking each individual one. And it makes it super easy to, for example, go through your time points here and just plot each individual one, because it's all considered like one thing, right? Okay. Questions? Anything you're interested in? Anything you would like to know and I haven't touched on? All right. I don't have anything else to add, so I'm just gonna reiterate that if you find yourself writing Python code for OMERO and you run into any issues, feel free to reach out to me. I am on ImageSC all the time. So feel free to post a question there. I will, if it's a question that I can address, I will show up to answer. But yeah, feel free to reach out to me in any shape or form. Yeah, thank you all for being here. This was fun. I like talking about this stuff. Yeah, hope to see you around at a conference or training or anything elsewhere. All right. See you all. Bye-bye.